First of all, ischemia occurs due to the interruption of blood flow. The most common and high-yield example of ischemia are infarction and stroke. The major concept of ischemic injury is that with decrease in blood flow, the delivery of oxygen and nutrients decreased. And without these two factors, tissue cannot maintain metabolism for a prolonged period of time. So what is the mechanism of ischemic injury? Let's take as an example myocardial infarction. With interruption of blood flow, the delivery of oxygen to the myocardial tissue decreases, and decrease in amount of available oxygen very rapidly forces myocardial tissue to generate energy by anaerobic glycolysis. Recall that usually organisms use aerobic glycolysis because by aerobic glycolysis, tissue produces 36 ATP molecules. Anaerobic glycolysis brings only 2 ATP molecules. But the anaerobic glycolysis is the only way how our organism can generate any energy in anaerobic condition. Anaerobic glycolysis has two major issues. First of all, anaerobic glycolysis causes lactic acidosis of myocardial tissue. And also because anaerobic glycolysis produces significantly lower amount of energy compared to aerobic glycolysis, ischemia creates an energy deficient state. So to understand how these two conditions occur, we will explain this step by step. This is the general picture of aerobic glycolysis that works in tissues that have enough supply of oxygen. In ischemia, the delivery of oxygen to the tissue decreases, and without oxygen, electron transport chain immediately shuts down because electron transport chain is not working in anaerobic condition. To explain why, recall that after complex 4 sucks the last portion of energy out of electron, complex 4 throws out this low energy electron from electron transport chain to oxygen inside the mitochondrial matrix. And electrons bind to oxygen and reduce oxygen with formation of water molecules. So what happens in ischemia is that there is no oxygen actually. And without oxygen, complex 4 cannot get rid of wasted electron. So electron stays in complex 4, thereby other electrons get stuck in electron transport chain. And while electrons are still in electron transport chain, complexes cannot intake new electrons. So these NADH and FADH electron transporters accumulate in the cytosol and just waiting around mitochondria until they will be able to donate electrons to electron transport chain. But the major problem here is that without transportation of electrons, complexes cannot generate energy to provide transportation of hydrogen ions into intramembrane space. So hydrogen-proton concentration gradient dissipates, and without hydrogen concentration gradient, ATP synthase cannot generate energy for phosphorylation of ATP molecule. So ATP production decreases, and with decrease in ATP production, myocardial tissue very rapidly is going into energy deficient state. Now to understand how lactic acidosis develops, we have to keep in mind that if electron transport chain is not working, NADH molecules cannot get rid of electrons, and this creates a huge problem. Recall that in glycolysis we have a reaction where glyceraldehyde 3-phosphate is oxidized to 1,3-bisphosphoglycerate in a reaction that is catalyzed by glyceraldehyde 3-phosphate dehydrogenase, and this enzyme as cofactor use NAD. Thereby this reaction results in formation of NADH, and in aerobic condition NADH delivers electrons to electron transport chain, and when NADH gets rid of electrons, NADH is converted into NAD, and now glyceraldehyde 3-phosphate dehydrogenase can catalyze another oxidative reaction. But in anaerobic condition, electron transport chain is not working. And the problem is that now without electron transport chain, NADH cannot get rid of electrons. But NADH anyway must be converted back to NAD because glyceraldehyde 3-phosphate dehydrogenase cannot provide oxidation without NAD. So as a result, NADH molecules progressively accumulate in the cytosol, and they have to find something to which they can donate these electrons. And the solution here is lactate dehydrogenase. Lactate dehydrogenase is an enzyme that is located in the cytosol, and this enzyme can convert pyruvate into lactate. Basically, lactate dehydrogenase catalyzes conversion of pyruvate to lactate and lactate into pyruvate. So this enzyme works in both directions. Everything depends on the amount of NADH and NADH in the tissue. So when NAD prevails, lactate is converted into pyruvate. If NADH prevails, pyruvate is converted into lactate. So in this case, lactate dehydrogenase is the only possibility for NADH to get rid of electrons. And NADH molecules see lactate dehydrogenase and pyruvate, they come to lactate dehydrogenase, and basically with help of lactate dehydrogenase, they massively donate electrons to pyruvate. That results in conversion of pyruvate into lactate, and simultaneously NADH are converted back to NAD. And now glyceraldehyde 3-phosphate dehydrogenase at least can convert glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. So this electrons donation to pyruvate with formation of lactate basically maintains activity of glycolysis, and this permits to generate ATP molecules in anaerobic condition. And as we see in anaerobic condition, pyruvate stays in the cytosol in form of lactate. And this makes sense because the function of the Krebs cycle is to produce NADH and FADH molecules that have to deliver electrons to electron transport chain. But in anaerobic condition, electron transport chain is not working. So basically, in anaerobic condition, Krebs cycle and electron transport chain become totally useless, and the entire energy generation process occurs strictly in the cytosol. The first problem with anaerobic glycolysis is that anaerobic glycolysis causes progressive accumulation of lactate inside the cell, and accumulation of lactate inside the cell causes decrease in intracellular pH, thereby leading to intracellular acidosis. And apparently, the longer is the period of ischemia, the more lactate molecules accumulate, the lower become intracellular pH, thereby the more severe becomes intracellular acidosis. Now, to understand why lactate causes decrease in intracellular pH, we have to know that actually the product of lactate dehydrogenase reaction is lactic acid. But in aqueous environment, as in cytosol, hydrogen ions easily dissociate from lactic acid, and this results in formation of lactate. So when we are talking about accumulation of lactate, simultaneously also accumulation of hydrogen ions occurs. So with anaerobic glycolysis, the amount of lactate and also the amount of hydrogen ions in the cytosol increase, and hydrogen ions make intracellular pH more acidic, or we can say that accumulation of hydrogen ions decrease intracellular pH and cause intracellular acidosis. So the first consequence of ischemia is intracellular acidosis. And also ischemia causes energy deficient state, because recall that glycolysis itself generates 2 ATP molecules. And in anaerobic condition, because electron transport chain is not working, 2 ATP molecules is all that we can produce. But if we have sufficient amount of oxygen, then pyruvate molecules are going into mitochondria, and degradation of pyruvate results in production of 34 ATP molecules. And 2 ATP molecules from glycolysis itself plus 34 ATP molecules that are produced in electron transport chain gives 36 ATP molecules. So in anaerobic condition, we can produce just 2 ATP molecules. In aerobic condition, glucose degradation results in production of 36 ATP molecules. So the difference is tremendous. That's why ischemia causes severe energy deficient state in myocardial tissue. Intracellular acidosis activates sodium-hydrogen exchanger that causes increase in intracellular sodium level. To explain this, recall that cardiomyocyte has sodium-hydrogen exchanger that regulates intracellular pH, and the accumulation of lactate with hydrogen ions induces activation of sodium-hydrogen exchanger. In response to decrease in intracellular pH, sodium-hydrogen exchanger begins to transport hydrogen ions out of the cell and sodium ions into the cell. This somehow corrects intracellular pH, but more important, this causes increase in intracellular sodium concentration. In addition to this, in ischemia, myocardial tissue is in energy deficient state, and without sufficient amount of ATP molecules, Active transporters that require ATP molecules for their function, they cannot maintain their normal transportation rate. So the activity of sodium-potassium ATPase in energy deficient state dramatically decreases. And now we have rapid income of sodium ions through sodium-hydrogen exchanger without the possibility to extrude them through sodium-potassium ATPase. And this causes progressive accumulation of sodium ions inside the cell. So energy deficient state disrupts the function of sodium-potassium ATPase, thereby creating very high intracellular sodium level. And high intracellular sodium level inhibits forward mode and induces reverse mode on sodium-calcium exchanger, and this causes dramatic increase in intracellular calcium level, a condition known as calcium overload. Recall that sodium-calcium exchanger can transport calcium in exchange of sodium in both directions, and the factor that determines in which direction sodium-calcium exchanger is working is intracellular sodium level. Sodium inhibits forward mode on sodium-calcium exchanger, and with huge increase in intracellular sodium level, sodium-calcium exchanger begins to work in reverse mode. And in reverse mode, sodium-calcium exchanger transport calcium ions into the cell and sodium ions out of the cell. As a result, calcium accumulates inside the cell. In addition to this, energy deficiency dramatically decreases the activity of SERCA protein. Recall that SERCA protein provides active transportation of calcium into sarcoplasmic reticulum, and active transportation requires energy. So in energy deficiency without ATP molecules, SERCA protein cannot provide transportation of calcium from the cytosol into sarcoplasmic reticulum. So now we have increase in calcium by reverse mode on sodium-calcium exchanger with decrease in calcium elimination from the cytosol, because SERCA protein without energy cannot work, and this causes massive increase in intracellular calcium level. So in addition to reverse mode on sodium-calcium exchanger, energy deficiency also impairs the function of the SERCA protein, and together this leads to a massive increase in intracellular calcium level. And calcium overload is a central event in pathogenesis of ischemic injury. Calcium overload causes activation of calcium-dependent proteases, so-called Calpain proteases. Activation of Calpain proteases causes increase in xanthine oxidase pathway that results in massive production of reactive oxygen species, thereby creating oxidative stress. To explain this, recall that in ischemia, the only possibility to produce energy is anaerobic glycolysis that results in production of only 2 ATP molecules. And 2 ATP molecules simply not enough to maintain entire cellular homeostasis. So ischemia creates energy deficient state where cell begins to use its own energy reserves in form of ATP molecules. In normal condition, when cell use ATP molecule, ATP is degraded into ADP and then to AMP, and then both AMP and ADP can be phosphorylated in oxidative phosphorylation and converted back to ATP. So by oxidative phosphorylation, cell restores its own energy reserves. But in ischemia, when ATP molecules undergo hydrolysis with subsequent formation of AMP molecules, they cannot be recycled back to ATP, because in ischemia without oxygen, lactin transport chain is not working. As a result, because cell cannot phosphorylate AMP back into ADP, AMP molecules accumulate inside the cell. Then AMP molecules are degraded into adenosine. Adenosine undergo deamination that results in production of inosine. Inosine undergo conversion into hypoxanthine, and hypoxanthine by xanthine oxidase is converted into xanthine. The degradation of xanthine to uric acid is provided by 2 enzymes, xanthine oxidase and xanthine dehydrogenase. These 2 enzymes have the same feature, they both convert xanthine to uric acid, but they do it in a different fashion. Xanthine dehydrogenase for conversion use NAD and water, and this results in production of two byproducts, its NADH and hydrogen ions. In contrast to xanthine dehydrogenase, xanthine oxidase use oxygen and water, and the byproducts of xanthine oxidase activity are superoxide and hydrogen peroxide molecules, which are highly active reactive oxygen species. So we see that xanthine oxidase pathway produce substantial amount of reactive oxygen species. In normal condition, the concentration of xanthine dehydrogenase is higher than the concentration of xanthine oxidase. And because of that, xanthine dehydrogenase pathway prevails over xanthine oxidase pathway. But in ischemia, calcium overload shifts hypoxanthine metabolism towards xanthine oxidase. And the concept here is that calcium overload causes activation of Calpain proteases, and activated Calpain proteases cleave a fragment of xanthine dehydrogenase enzyme, and this cleavage converts xanthine dehydrogenase into xanthine oxidase. Apparently, the level of xanthine oxidase increase and the amount of xanthine dehydrogenase decrease. Thereby, during ischemia, xanthine oxidase pathway prevails over xanthine dehydrogenase pathway, and this results in increase in production of reactive oxygen species that cause oxidative stress. Also, in activated state, Calpain proteases begin to hydrolyze cytoskeletal proteins, as vinculin for example, and this causes damage to sarcolemma of cardiomyocytes. Sarcolemma becomes more fragile, and it greatly increases the risk of sarcolemma rupture. And in addition to this, calcium overload causes abnormally strong myocytes contractions. Recall that with increasing amount of calcium molecules in the cytosol, more calcium molecules will bind to troponin, so the stronger troponin will affect tropomyosin conformation, the higher will be the force of contraction. In normal condition, it's called positive inotropic effect that helps heart to pump the blood. But in ischemia, calcium concentration inside the cell becomes so high that it becomes dangerous. There is enough when there is not too much, and in state of calcium overload, there are a lot of calcium molecules in the cytosol that cause abnormally strong myocytes contractions. And the concept here is that in fact, every contraction of the cardiomyocytes is a physical stress for sarcolemma, and the higher the calcium overload, the stronger become contractions. The stronger are contractions, the more severe is the physical stress on sarcolemma, so the higher is the risk of sarcolemma rupture. And as we know, in case of ischemia, sarcolemma is already predisposed to rupture due to the Calpain proteolytic activity. So damage to the cytoskeletal proteins and abnormally strong myocytes contractions greatly increase the risk of sarcolemma rupture. Also, calcium overload induces opening of the pores on inner mitochondrial membrane, the phenomenon called mitochondrial permeability transition. Mitochondrial permeability transition leads to activation of intrinsic apoptotic pathway that results in cellular death. Now, the concept is that because mitochondrial matrix has a higher proteins concentration than the cellular cytosol, once membrane becomes permeable, fluid is going by oncotic gradient from the cytosol to mitochondrial matrix. This causes mitochondrial swelling that at some point results in rupture of both mitochondrial membranes. And with rupture of the outer mitochondrial membrane, cytochrome C is released into the cytosol, and cytochrome C induces intrinsic apoptotic pathway. At the same time, increase in reactive oxygen species induce severe oxidative stress inside the cell. First of all, oxidative stress causes severe oxidative damage to plasma membrane. Basically, free radicals cause oxidation of lipids of the plasma membrane. This results in substantial damage to sarcolemma, and the damage to sarcolemma substantially increases the risk of sarcolemma rupture. Also, reactive oxygen species cause damage to the DNA molecule, and obviously all these processes lead to a cellular death. So, ischemia induces three major pathological conditions. It causes intracellular acidosis that leads to a calcium overload, and calcium overload causes oxidative stress. What is important to understand is that during the first 20 minutes of severe ischemia on its initial stage, all these processes can be reversed. But if ischemia is prolonged, it induces such lethal events as, for example, mitochondrial permeability transition, which is the point of no return, and irreversibly lead to a cellular death. So prolonged ischemia results in cellular death, and the three crucial pathological events are intracellular acidosis, calcium overload, and oxidative stress.